Why G6PD deficiency is more commonly seen in malaria-endemic region. Why G6PD deficiency is particularly more common in certain parts of the world, like sub-Saharan African region or Mediterranean region or certain part of the Asia, Southeast Asia. G6PD is seen in all kind of cells, but why G6PD deficiency particularly present with hemolytic anemia, particularly involving RBC? Why the hemolysis occurs in the G6PD deficiency? Why the G6PD deficiency patient, when you do the testing, it usually comes falsely negative during the periods of hemolysis. All this and much more we are going to discuss in this 98th edition of the InstaLecture on the G6PD deficiency. So G6PD deficiency, by the way, is the most common enzyme deficiency in the world. It's the most common enzyme pathology affecting 400 million people worldwide. That is a huge number of people. Thinking that 7.5 billion people, roughly, we are living in the current planet. Now, I would start with the first question, which has been asked to me, that what is the connection between the malaria-endemic region and the G6PD deficiency? Curiously, it has been noted since middle of the last century, a lot of researchers note that the patients who are having G6PD deficiency, they are more commonly seen in certain pockets of the world, particularly sub-Saharan Africa, in the northern African region beneath the Sahara, and that Sahara Desert. And that part of the world, the malaria is very much common. So the current idea is this, it is thought in this way, that as those part of the world, the malaria is very much common, a common cause of death, certain kind of evolutionary genetic protective mechanism, this thing has occurred, that the G6PD deficiency is seen in the RBC. So if G6PD deficiency is seen in the RBC, there will be more free radical generated in those RBCs of the patient. And due to the presence of those free radicals due to the presence of this oxidative stress in the RBCs, it makes much more difficult for the Plasmodium species to invade and flourish in those RBCs. So basically, it becomes more difficult for those parasites to survive in the RBC. So it kind of protects against the malaria infection. So in those parts of the world where there's a large number of people that die due to malaria, in those parts of the world, it has been seen that as a kind of protective mechanism, the RBCs have developed some kind of G6PD deficiency. And why I already explained, because if there is G6PD deficiency, there will be more free radical, more oxidative stress in those RBCs, and that would create some kind of hostile environment for those parasite Plasmodium species to thrive and survive. So in a way, it protects. So that's why those parts of the world where this Plasmodium species, that is malaria, is very common, like certain parts of the Southeastern, Eastern Asia, certain part of the Northern Africa, Sub-Saharan African region, this disease is, this G6PD deficiency is much more prevalent. Now, G6PD deficiency first got attention, particularly when certain group of American soldiers, they basically went to the war field, and when they are taking anti-malarial drugs, because those, as they were went outside, went abroad, went overseas, and those part of the world they went where there is a lot of malaria, malaria endemic area is there. So they used to take anti-malarial drug like primaquine, and they were developing acute episodes of the hemolysis. So they, at that time, actually, it got attention that these people, they're developing actually the acute hemolysis, and they found that there's some kind of missing connection is there, that why they're developing the hemolysis after having antibiotic drug. And then they found out that this is usually that these people and these soldiers, these American soldiers, are mostly from Mediterranean descent and Afro-American origin. So particularly this group of soldiers, American soldiers, they were developing, and that's how they found out that there is some kind of connection between drug-induced hemolysis, anti-malarial drug-induced hemolysis, and this is another part of this thing, that how the G6PD deficiency and its manifestation came to our attention in much more. But already I had answered the first query, that why the malaria endemic area the G6PD deficiency is seen as a part of kind of, you can think in a kind of genetic evolution where making the cells less G6PD, making less G6PD available in those RBCs and generating some kind of more pre-oxidized radicals. The nature is making it hard or difficult for the Plasmodium parasites to survive and thrive in those RBCs, and making it difficult to infect actually, or to cause severe complication in those patients in those parts of the world where the malaria is very much common. Now, G6PD deficiency is the other key concept that you need to know, that basically it's a X-linked recessive disorder. So particularly it manifests in the males, and the females are carriers. And it's usually seen in the long arm of the X chromosome in nearby to the area of other key genes, causing other diseases like hemophilia A or color blindness. So basically it's seen in the long arm of the X chromosome, and it's a X-linked recessive disorder. So particularly, mostly the males would be the patients and females would be carriers. What would be the manifestation of this disorder? Manifestation of this disorder could be mostly the presence with acute episodes of episodic hemolysis when they are exposed to certain infection or certain drugs like nitrofurantoin, ciprofloxacin, certain NSAIDs, and obviously certain antimalarials like primapine. Or another cause is FAVISM, that if they're eating, so mostly common seen in the young children, they're fava beans, which is particularly commonly eaten in certain parts of the world like Mediterranean region, along with soup or salad, or sometimes they usually take the fried fava bean as a snacks also. Particularly is more common in the certain Eastern Asian population. They usually take it as snacks. So when they consume those fava beans, that also causes oxidative stress and can cause episodes of acute episodic hemolysis. And particularly this FAVISM is very much common. It could cause severe, basically, G6PD deficiency in certain individuals. And also G6PD deficiency can present with chronic hemolytic anemia, neonatal hyperbilirubinemia, in certain percentage of cases. And most of the patient, by the way, though they present as asymptomatic, there is no presentation. They never knew. They sail through the life without knowing that they're having the G6PD deficiency smoothly. They sail through their life smoothly without being unaware of this condition that they're affected by this G6PD deficiency. So now I would try to go to the whiteboard and try to explain that why the hemolysis occurs. I would try to make it simple, the concept simple, that you should understand that why this hemolysis is occurring. So the idea is very simple, that this RBCs, they basically live in an environment of oxidative stress. So as they live in an environment of oxidative stress, due to the oxidative stress means they actually, in the RBCs, there could be generation of the free oxygen radicals like H2 or other free oxygen radicals. And this could occur due to many reasons, because as there is continuous inflammation going on the body of the macrophages, when you are taking certain drugs, they can produce this H2O2 or other free oxide radicals. Now, actually, normally what happens, this H2O2 is neutralized, or these free oxide radicals are neutralized by the reduced glutathione, GSH. And this GSH basically becomes GSSG, that is oxidized form. So this reduced glutathione is very much important for basically for the survival of the RBCs, because this GSH protects RBC from the oxidative stress or oxidative damage. Now coming back to this G6PD enzyme, this G6PD enzyme is basically it's a very rate limiting enzyme in the hexose monophosphate shunt, HMP shunt, which you already know. So this G6PD, what they do, this G6PD enzyme through HMP shunt pathway, they basically produce NADPH. And this NADPH is very much crucial because this NADPH is basically involved in production of this GSH. To maintain continuous production of the reduced glutathione in the RBC, they require NADPH. And this NADPH is produced by the hexose monophosphate HMP SUNT pathway through this enzyme G6PD. That is the core part. Now, the question is this, that why RBCs are particularly vulnerable for the G6PD deficiency? Why all the cells in the body, they're having G6PD deficiency? Why the problem is only affecting RBCs? And there are particularly two reasons behind this. Because firstly, that for the RBC, the sole source for the NADPH production is this G6PD HMP SUNT. They don't have any other way. So obviously, if the G6PD enzyme is not there, that means glucose 6-phosphate is not, while they're converted to 6-phosphoglucone. That step, the G6PD, when there is not there, obviously that would not produce the NADPH. And if the NADPH is not there, the reduced glutathione cannot be generated. So, and the second thing that also you need to think in this way, that if you think that RBC is basically a very simple biological cell, RBC has no nucleus, has no mitochondria, they don't have any ribosome also, because they basically want to make a simple cell which contain only and only hemoglobin. So in the mature RBCs, the ribosome and the nucleus is not there. So in other cells, actually, later, due to the presence of nucleus, ribosome, they can produce even other proteins. But if RBC want to produce, mature RBC want to produce proteins like G6PD, they cannot produce it. And more importantly, as I mentioned, that NADPH, the sole source of NADPH in the RBC is this hexose monophosphate pathway involving G6PD. So if it is not there, if it is deficient, definitely they don't have NADPH. So when they don't have NADPH, don't have G6PD, they don't have NADPH, they cannot produce NADPH, and they cannot produce reduced glutathione. When they cannot produce reduced glutathione, obviously what would happen? These free radicals, they would damage the RBC membrane. They would damage the RBC membrane, and they would also denature the hemoglobin, which would precipitate nearby the membrane. These are usually called hinge bodies, and this could be detected also microscopically. I'll be discussing about this earlier and also discuss in a separate session, because in this condition we usually see hinge bodies, and also we see bite cells and blister cells, but that is a topic of separate discussion. So net result is this, that for the survival of the RBCs from the oxidative stress, the RBCs require reduced glutathione. When the reduced glutathione is not there, they become vulnerable to be damaged by oxidative stress. And oxidative stress is usually caused when you usually suffer from infection, or usually taking certain drug, or fava beans, which are exposed to FAVISM, fava beans. In these cases, oxidative stress occurs. Usually the macrophages produce more oxidative radicals, and that is caused the problem. Now, in this case, what's happening, that if you don't have G6PD, you cannot produce NADPH, because this is the sole source for NADPH production in the RBC. And you cannot produce NADPH, you cannot regenerate production of reduced glutathione. And when you cannot produce reduced glutathione, what would happen? The RBC would be continuously damaged by the free oxide radicals like H2O2, and that would cause membrane damage of the RBC. That would cause denaturement of the hemoglobin, particularly of the globin, and then they would be deposited as hinge bodies. And obviously these cells could undergo hemolysis, and in G6PD deficiency, both intra-vascular and extra-vascular hemolysis could occur, ultimately resulting in hemolysis or the development of the hyperbilirubinemia and jaundice. And these episodes usually occur, typically in most of the patient, as after exposure to certain infections, certain drugs, certain chemicals like naphthabol or 2-TNT, trinitrotoluene. These chemicals can also cause, naphthabol is very much important. It can cause this, and obviously certain drugs which I mentioned. But usually when the patient come to know that they are G6PD deficient, they avoid those drugs. So in those cases, the more important cause would be usually infection. Infection followed by infection triggers these episodes of the episodic hemolysis. Now, the other aspects that you need to keep in mind about the G6PD deficiency, that G6PD deficiency, if you do in the acute episodes of the hemolysis, if you want to check the G6PD level, often the result would come negative. That means they would result to show that this patient is having adequate G6PD. The reason for this is that when you have acute episodes of hemolysis, usually a lot of reticulocytes come into the circulation. And the reticulocytes usually have higher level of G6PD. So that is not the right time during the acute episodes of the hemolysis to do G6PD testing. You should do it usually after a period when these old RBCs are available. And at this, another reason would be the result would be negative because those RBCs which are lacking the G6PD, this particularly the old RBCs, they're mostly destroyed by that time. So obviously you will not get a positive result. So that is not the exact time to do G6PD testing. And ideally you should do after a few weeks. Best to do after actually 3 months, because after usually they leave for 120 days. That's after that period you'll be getting a better accurate result actually, G6PD testing. And obviously G6PD by WHO, they have been categorized into 5 grades: grade 1, grade 2, grade 3, 4, 5 actually. The 1 is usually the most severe and they can present with congenital hemolytic anemia. Grade 2 and 3 are usually present with episodic hemolysis, but 2 is quite severe, and 4, 5 is clinically not significant. That is the part you need to do. And obviously the diagnosis and other part I would do a separate discussion where I would compare with pyruvate kinase deficiency and other hemolytic anemias. So in a nutshell, what we learned in today's session on the G6PD deficiency, that this is the most common enzyme pathologies in the world, affecting 400 million people. G6PD deficiency is particularly more common in certain part of the world where there's malaria, those part of the world which is affected or hit badly by the malaria, parasitic infection. And why I told you that if the G6PD is less available, there is more free radical generation within the RBC, and in that kind of environment with a lot of free radicals, it is difficult for Plasmodium parasites to survive. So this is one of the idea, the thing that why in those part of the world G6PD deficiency is more common. So it's a kind of protective mechanism, evolutionary protective mechanism you can think in this part of the world. They are particularly seen in more common in certain parts of the world like sub-Saharan Africa, certain part of the Africa. They're also more common in the African origin people, African descent, Mediterranean region people, and also Southeast Asia. The next part I told you that this is actually an X-linked, this is the disorder. And the other key concept that they usually can present with acute, mostly the patients remain asymptomatic, they can present with acute episodes of hemolysis, usually followed by certain triggers like infection or exposure to certain drugs or certain chemicals or fava beans. When the young child eat a lot of fava bean containing food, it may be a salad, maybe a soup or some meat preparation or some snacks which contain the fava beans. And obviously after this, they usually can develop some episodes of hemolysis, jaundice, back pain, and dark colored urine. This is the typical. In some patient, there could be a chronic hemolytic anemia could also be seen in some severe cases, but mostly the patients remain asymptomatic. And in the symptomatic patient, most common presentation is acute episodes of episodic hemolysis, acute hemolysis, hemolytic anemia. And then I may explain to you that why the hemolysis occurs in the patient of the G6PD deficiency. Why RBC is only affected? RBC is only affected, as I mentioned you, that RBC has usually no Firstly, the sole source of NADPH production in the RBC is this HMP shunt pathways. So, and HMP shunt pathway, the key rate limiting enzyme is the G6PD. And this actually, through this pathway, the RBCs are producing NADPH. And NADPH is required to produce reduced glutathione. So when your RBC is G6PD deficient, they cannot produce NADPH. When they cannot produce NADPH, they cannot generate reduced glutathione. When you don't have reduced glutathione, which is the key protective chemical molecule from the attack of the oxidative stress, the RBC becomes more vulnerable to be attacked and damaged by the oxidative stress. So their membrane could be damaged. They could be, the hemoglobin could be denatured, and all these things could precipitate causing hemolysis. That's why hemolytic anemia would be seen, which can present with jaundice, which can present with obviously anemia, usually the acute anemia. But as I mentioned, in certain patients, they can be present of chronic hemolytic anemia or neonatal hyperbilirubinemia could be present. And so that's all this session on this G6PD deficiency. The other aspect of the G6PD deficiency, particularly the white cell, the hinge body, and the blister cell, and the other diagnostic aspect are discussing in the session. So thank you very much for attending this session. Have a nice day.